Thank you, thank you. I'll try to make sure whoever's in here last leaves it open for y'all. Should he come like this? They might not be logged in just yet, but I'll be up there if something happens, let me know.
Okay.
All right, thank you.
Thank you.
Okay, have a.
Is Miss is Miss Warden back this week?
I believe so, yes.
I couldn't remember if she said just last week or this week.
Okay, let's get started. Can y'all see me in this time?
I'm sorry, what was that?
Can y'all see me on both boards?
No. You should.
On the bottom of the screen, on the little white screen,
Yes, ma'am.
click, can't remember what it's supposed to say. Hang on just a second, let me see. Do you see briefing room?
Yes, ma'am.
Click briefing room and then click discussion.
Okay.
That help?
Yes, ma'am.
Okay. And I think y'all will have to do that every time in order for me to be on one screen and then, there we go. I see y'all. Okay. I had another teacher ask me today how my high flex class was going, and I told him it's working great. It's kind of like y'all are right here with us, and they were like, well, my students at Forest County won't participate in discussions, and so I was able to say that y'all do a good job of participating just like you're here. So we actually are sitting here right now, and we see y'all back there on the back screen, so it really looks like y'all are right here with us. So I think this is working out great. Okay, let me call attendance real quick. I think we got everybody. And then we'll get started. All right, Anna. Jacob.
Here.
Okay. Honor. Sean. Ryan. Seth. Cameron is out. I want Cameron. Abby. Brooklyn. Kaylee. And Brianna. Okay. And then Forest County, Kristen.
Here.
She.
Here.
And Rachel.
Here.
Good. Okay. All right. This week, we're going to pick up in Module 5, I think, is where we are. Let's see. color. Yeah, we're in 5. Okay. So everybody should have taken test 1, either here or in Forest County last week. Had pretty good results. Had several A's, so that was a good thing. And then on test 2, we're going to cover chapters 4, 5, 6, 7, and 8. Okay, so this is a big portion of the material will be on test 2, and we'll kind of consider that our midterm, even though it might not technically be right at midterm. Okay, so the next test is going to be out a little while. We're going to start chapter 5 today, and then we'll move into 6, all about cells. 7 will be about cell transport, and then 8 will be about metabolism. Today is chapter 5, which is all about macromolecules.
I have a question of the test, question 12, which is when you type official genus and the species name of an organism, what do you do then? I choose like all of them.
Okay, so if you're going to type the genus and species name, that goes back all the way to chapter 1. The genus will be capitalized and then the species will not. And that will be, if you're typing it, then you would put it in italics. If you're handwriting it, then you would underline it.
Oh, underline. So
Yeah.
the correct answer is underline them, right?
Did the question say if you're typing it or if you're writing it?
The question say, what do you do to them?
If what?
So there is a several options.
When do you type the official genus and species name for an organism? What do you do to them?
Okay, what's our choices?
You can bold and underline them, capitalize both of them, underline them, or italicize them. I'm pretty sure it's italicize.
That's right.
Yes. You would put it in italics if you're typing it, and if you were handwriting it, then you would underline it. So that would let people know that that was an official name of something.
Okay, I got you.
Okay? Hey, Cameron.
Hi.
Okay. All right, so we're getting started in Module 5 with Chapter 5, which is all about large biological molecules. There's that word again, bio. What do we say bio means? Life or living. Okay, so these molecules are going to be molecules that we are going to find in living organisms. Okay? Let's zoom in so we can make our slides a little bigger. Right on them. So it says macromolecules. First of all, let's make sure we get this term. Everybody write this term down. Macro. What does micro mean? Micro means small. So opposite of that is macro. And we're talking massive, okay? Very, very large. So these are going to be really large molecules. And so macromolecules are going to be considered polymers that are built from monomers. So that's going to be an important part of understanding macromolecules. Kind of like Legos. If we were going to take and build a big castle out of Lego blocks, the whole entire structure would be our polymer. Each individual little Lego block would be a monomer. Okay? So we can kind of imagine macromolecules being the big entire project, and each macromolecule is going to have its very own specific monomer that we're going to use to build our big polymers. It says large polymers are known as macromolecules for their huge size. They're really, really big. Okay, so what is a polymer? Get this definition. A polymer is a long molecule consisting of many similar building blocks. So these building blocks are going to repeat themselves over and over and over. Polymers are long molecules consisting of many similar building blocks. The repeating units that we're talking about that are going to be the building blocks, so they're going to serve as building blocks, those are called monomers. So technically we could say monomer plus monomer plus monomer will give us the entire polymer. Okay, so polymers are made or built from monomers. And so the macromolecules that we're going to talk about today, carbohydrates is going to be one of them, proteins, and nucleic acids. And we'll talk about another one called lipids. They are going to be giant. Okay, they are big macromolecules, and they're going to have their own individual monomers. Okay, so monomers build polymers. We're going to talk about two ways or two reactions that can happen. One will be building big polymers, and then one will be breaking down big polymers. Okay, is everybody caught up? And so it says three classes are macromolecules that are polymers. We're going to talk about carbohydrates. Y'all can list these. Y'all can just say, let's just say 4 macromolecules. We have 4 macromolecules. We're going to talk about carbohydrates, proteins, lipids, and nucleic acids. Okay. Lipids are not technically going to be called polymers, but they are going to have a base unit that they're going to be made of. Okay. So these 4 big polymers all have a monomer. So in this picture right here, this is showing this big long chain. They're calling it a polymer. Each individual unit is the monomer. Okay? We'll come back and we're going to make us a little chart to kind of help us break all this down. It says the synthesis, remember synthesis, that word means to build. So the synthesis and the breakdown of polymers. We're going to have 2 reactions that we're going to talk about. And in order for these reactions to happen, we're going to use the help of enzymes. So what is an enzyme? Get this definition. It is a specialized macromolecule. So it is actually a protein. Okay? A specialized macromolecule that will speed up chemical reactions because it's going to lower the activation energy and allow the reaction to happen much quicker. And so enzymes are used in making and breaking the bonds between monomers that build polymers. Okay? All right. Here's your next thing I want you to say in your notes. We're going to kind of build a little outline here. The next thing I want you to say, we've got 2 types of reactions. Two types of reactions. Number 1, we're going to let that be dehydration synthesis reaction. Right here it just says dehydration reaction. You may see it both ways, but it's also called a dehydration synthesis reaction. Remember we said synthesis means build. That's going to happen when 2 monomers bond together by losing a water molecule. Okay? Think about what the word means. Dehydration, that means to take away water, right? And synthesis means to build. So we're going to pull out a water molecule to build a big giant macromolecule. Number 2, our second reaction, is going to be called hydrolysis. And if we break this word down, hydro means water. So hydro means H2O, water. Lysis means to break. So in a hydrolysis reaction, it's going to be the reverse. This is where we are going to add water, and we will add water, add H2O to break. So we're going to break down large molecules by throwing in a water molecule. And y'all know that little saying, you are what you eat? You ever heard that? It's true, okay? So if you are needing to build proteins, what do you want to eat a lot of? Proteins, right? Okay, so if we're needing to build proteins for muscle structure or for antibodies or for enzymes, you're going to want to have a good, healthy diet of proteins. If you're going to go hiking today and we're going to need a lot of extra energy, then you're going to want to eat a good diet of carbohydrates, okay? Because you're going to need to build some energy, okay? And so that little saying, you are what you eat, is really true. All right, so let's look at a picture of what we just talked about. We said our first reaction was called dehydration synthesis reaction. We said we could just look at the term. Dehydrate means to take out of water. Look at this water molecule right here. Y'all see this? There's the hydrogen, and there's the OH. If we put those together, that builds that water molecule. If we pull that out of these two monomers up here, then these two monomers can be joined across here like they are in the picture right here. Okay, so now we have a longer polymer because we've removed the water molecule, which allowed the monomers to stick together. Okay, so we're actually creating a little bond right there between those monomers. Okay, with hydrolysis, we said hydro means water, lysis means to break. So now we can throw in a water molecule. Here's the water molecule. We're going to put part of it over here, part of it over here. It will separate the bond that is normally between those two monomers and break them apart. Okay? So if we started with this big giant polymer, we could break this one over here. We could break it off if we added in a water. Okay? So you've got to stop and think about those terms, and it'll help you to understand their definition. Dehydration synthesis, allow the water to build. Hydrolysis, add a water to break. Okay? So these little definitions right here are good. Allow the water to build, add water to break. Okay? So it says the diversity of polymers. A cell has thousands of different macromolecules, so you're needing to build and break these bonds all the time. Macromolecules vary among cells, so different cells need to build different macromolecules. And then it says that vary within species and even more between different species. A huge variety of polymers can be built from a small set of monomers. One example is kind of like the alphabet. How many letters do we have in the alphabet? 26. Okay. How many words can we build? A lot. Thousands and thousands of words, right? Well, that's how some of these polymers are. We can take a few small sets of monomers, like proteins, for example. We have 20 different amino acids that are going to be used to build thousands of proteins. Okay? This tells us to watch the monomers versus polymer video. So let's go see our video here. I can get it. There we go.
Hi, this is Mr. W from ScienceMusicVideos.com, and this video is about monomers and polymers. Big biological molecules like DNA are built from smaller building blocks called monomers. Let's look at a big molecule that we eat a lot of, starch. Here it is. Notice this repeating unit. It's glucose, and you can see that starch is just one glucose chained to the next over and over again. That makes glucose the monomer, while starch is a polymer, a molecule composed of linked monomers. To remember this, use the prefixes. Mono means one, as in monorail. Poly means many, as in polygon. Think about it in terms of Legos. Individual Lego bricks, those are monomers. The structures you build by putting Legos together, those are polymers. All life is built from a relatively small number of monomers that combine into an astoundingly large number of polymers. Just think of our own bodies with a variety of proteins like muscle, or oxygen-carrying hemoglobin, or thousands of enzymes and millions of antibodies. We build all of these from 20 protein monomers called amino acids. It's just like the alphabet of any language, like English. You can take 26 letters, which are like monomers, and combine them into hundreds of thousands of words, which are like polymers. In cells, protein catalysts called enzymes take monomers and chemically link them together. It's called dehydration synthesis. The enzyme pulls an H, a hydrogen, from one monomer and an OH, a hydroxyl, from the other monomer, and links them together at that point. The H and OH combine to form a molecule of water. That's why it's called dehydration synthesis. Here's a sweet example. Here are two carbohydrate monomers, glucose and fructose. Through a dehydration synthesis reaction, they combine to form sucrose, or table sugar. Organisms also need to be able to break polymers apart. It's a big part of what happens during digestion. Enzymes break the bonds between monomers by jamming a water molecule in between them. It's called hydrolysis, splitting with water. Of course, monomers can be further broken down. Later on in this course, we'll look at cellular respiration, in which cells take glucose and break it down all the way to carbon dioxide and water as cells make ATP. In addition, monomers have to be made. Later on, we'll see how during photosynthesis, chloroplasts can take carbon dioxide and water and combine them to form glucose, which in terms of matter and energy is the starting point for just about everything else. So that's it for monomers and polymers. Right now, go to ScienceMusicVideos.com.
Okay? All right. Polymers are big. Monomers are the building blocks. We got two reactions that make and break those big molecules. Make sure you know those. Okay? All right, we're going to start in section 5.2 with our first macromolecule. Okay, so we're going to kind of start with carbohydrates first. So number one, carbohydrates. These are going to be what we call the sugars. And sugars are going to give us quick energy. And if you've ever been around small children, if you feed them a bunch of sugar, it works pretty fast. Okay? Especially take some of them trick-or-treating and let them start digging in their candy bag. And you will not want to be around them very long. Okay? They will get very hyper. They got all this little extra energy wanting to run around all over the place. Okay? It hypes them up. We do this if we were playing a sport. You may want to drink a sports drink, a Gatorade or Powerade. They have lots of sugars to give you some quick energy. Okay? It says the simplest carbohydrates, so the smallest sugars, we're going to call those monosaccharides. So our first one is going to be monosaccharides. Let me skip ahead for a second. We'll come back to that on the next slide. So hang on just a second. Monosaccharides are going to be very simple. It says carbohydrate macromolecules are polysaccharides, and they are going to be composed of many sugar building blocks. Okay, so our main idea there from this slide is that carbs give us quick energy. Let's start with our first carbohydrate. They are the monosaccharides. So anytime you see this root word saccharide, you're thinking sugar. Also, sugars are going to end in OSE. We're going to talk about glucose and fructose and sucrose. So those words that end in O's, you know those are going to be carbohydrates also. Okay? Monosaccharides are going to have a carbon to hydrogen to oxygen ratio of 1 to 2 to 1. For every 1 carbon, there will be twice as many hydrogens to 1 oxygen. Okay? Carbon, hydrogens, and oxygen. One example, our number one example, is glucose. It is what we call our simple sugar. It is the most common monosaccharide. Its formula, go ahead and write it down because we're going to talk about glucose in every single chapter from here on out. C6H12O6. Do you see how if we were to pull out the ratio, you've got 6 carbons, 12 hydrogens, and 6 oxygens. So that's a 1 to 2 to 1 ratio. Okay? So it says they're classified by the location of the carbonyl group, then the number of carbons in the carbon skeleton. Here's some common examples over here of some monosaccharides. Okay? Some of the ones that we're definitely going to talk about is ribose. We'll talk about that when we're talking about ribonucleic acid. So you've probably heard of ribose being part of your RNA. Fructose, that's that sugar we talked about getting from fruit sugars. And then if you know anybody who's a diabetic, you know they wouldn't be able to break down glucose without the help of taking insulin. Okay, so mono means 1, saccharide means sugar. So our simple sugar, number one example is glucose. You could add fructose there if you want to. Okay. It says, though often drawn as linear skeletons, these aqueous solutions, in aqueous solutions, many sugars form rings. So they can form straight chains, they can be branched, or they can form rings. This is monosaccharides serve as the major fuel for cells. So we said that these are going to give us quick energy. It's kind of like putting gasoline in your car to make it go down the road. Your cells are going to require glucose in order to do their daily functioning. Have you ever gotten a stomach virus and you're not able to hold down foods? Can you still perform all of these daily activities? No, very quickly you'll find yourself having to lay down because you can't even hold your head up. Okay, so your cells, if they're not able to take in these carbohydrates, then you run out of energy very quickly. Okay? Our next carbohydrate. So right now we've got monosaccharides. Next is disaccharides. For math class, what does di mean? 2. What do we say saccharide means?
Sugar.
Sugar, good. So 2 sugars. And this is going to happen when we join 2 monosaccharides together. It says this covalent bond between 2 monosaccharides is called a glycosidic linkage. So if we look at our pictures here, and this kind of goes back to our reactions we talked about earlier. In this reaction, we're going to build maltose. And maltose is a disaccharide. Okay, so this is the disaccharide. If we look here, here's the whole entire thing. Okay, we would technically consider it on its way to be a large polymer. And it is made by taking glucose number 1 and adding it to glucose number 2. Okay, so if we take 2 glucose molecules, if we take the hydroxyl group off of this molecule over here, and we take a hydrogen off of glucose number 2. Do y'all see how those two can build a water molecule? If we pull the water molecule out of this reaction, if we dehydrate it, that's why it's called dehydration, then we can link those two together right here and form a disaccharide. Here's a common one, sucrose. Everybody likes white table sugar, right? You like to put that in your tea or your coffee maybe. That is made by adding a glucose, which is a monosaccharide, plus a fructose, which is that fruit sugar we talked about. It's a monosaccharide. So 2 monosaccharides put together, glucose and fructose, can build sucrose. Okay? Dehydration synthesis, pulling out water to build. Our next carbohydrate is called a polysaccharide. Poly from math class means what?
Many.
Good. So many.
Sugar.
Many. And saccharide means sugars. Good. So many sugars all chained together are going to build polysaccharides. It says the architecture and function of a polysaccharide is determined by the sugar monomers and the position of its glycosidic linkages. So we're going to talk about 4 large polysaccharides. Okay, so title it 4 large polysaccharides. And remember, they're going to be made from building these monosaccharides together through dehydration synthesis reactions. All right. Number 1. So this is number 1. This is, we're going to talk about 4 polysaccharides. So this will go under polysaccharides, okay? Number 1 is starch. This is a storage polysaccharide of plants. And it's going to be made from linking lots of glucose molecules together. Give yourself an example. When that air conditioner cuts off, I'm screaming at y'all. An example would be a potato and a potato plant. So a potato plant, we know it's got the greenery up above the ground. The sun shines on it. It takes in water from its roots. It pulls out carbon dioxide from the atmosphere to build sugars. Okay, and it's going to use these sugars to build leaves, stems, and roots. However, once it's finished building all of these things, the sun's still shining. It's still got plenty of water. It's still able to pull in carbon dioxide. So it's going to continue to make sugars, but it's going to store them up in the form of a potato in the bowl. So it sends all of these extra sugars down to the potato. We dig it up. We go bake it, or we cut it up into French fries, or we make potato chips out of it. When we're doing this, what are we eating lots of? Starches, which are large sugars that we have to break down to get the monosaccharides out of. Okay, and so the monosaccharide would be glucose. So you know that if you eat a potato, 1 baked potato is the same as eating about 1/2 cup of white table sugar. Okay, and so you get lots of calories out of eating potatoes. So if you sit around, you've heard that term, a couch potato, sitting around eating a bunch of potatoes, if you're not burning that off, what will our bodies do with it?
Store it.
We'll store it up, okay? So we have to be careful when we're eating a bunch of these large starches. Where else can we get starches? Rice, pastas, breads. Okay. These are all very tasty. However, we have to be burning that off or we will store it up. Okay, if you are wanting to lose weight, you can cut out some of your carbohydrates and very quickly, as long as you kept exercising, you very quickly see that you could lose some weight that way. Okay? I think we already talked about all of those. All right. Number 2. Our second big polysaccharide is called glycogen. This is a storage polysaccharide for animals. So starch was for plants, glycogen will be for animals. Like us, we're animals. All right, let's pretend we went this morning and we had breakfast, okay? And we went all out. We had some biscuits and we had some eggs. We had some syrup and pancakes. We had an orange juice. Okay, we had lots of carbohydrates. Okay? Are we going to use all of those right then? No. Okay, that's way more than what we need first thing in the morning. Now let's pretend that we weren't able to have lunch. We've had class. We've got a test this afternoon. We're not able to have lunch. And guess what? You get to the cafeteria this evening and the cafeteria's closed. You don't get to have supper. Are you going to die? No. Not today. Okay, here's why. A bunch of these carbohydrates that you've been consuming that you didn't need are going to be sent to your liver, and they're going to be linked together to build glycogen, and they're going to be stored there in your liver. And so all throughout the day, little bits of sugar are going to be released back out into your bloodstream, and you're going to have glucose for the rest of the day because you kind of banked it up in the form of glycogen earlier that day. Okay? And so it is a storage polysaccharide. We store it in the liver. You need to know this. That's a test question. Where is glycogen stored? In the liver, and sometimes in muscle cells, mostly in the liver. It says hydrolysis of glycogen in these cells, so when we break it down, it's going to release it. It's going to release that glucose whenever you're needing it throughout the day. That's a good thing. Kind of like a little sugar storage for animals. Okay? Everybody good? All right, number 3. Well, let's talk about the pictures first. Here's a couple of pictures. Here's a picture of starch. Remember we said that's from plants. It's made by linking together glucose plus glucose plus glucose plus glucose. It may be 1,000 glucose monomers long, okay? Really, really large molecule. Here's glycogen. Same thing again, except this is in animals, and your liver will store all of those extra glucose molecules, link them together, form glycogen, and then use them throughout the day. All right, number 3. Number 3 is cellulose. There's that root word I was talking about. Ose lets you know that it's a sugar. It says the polysaccharide cellulose is a major component of tough cell walls of plants. They're going to make up the cell walls of plants. It's also going to be a polymer of glucose, so these glucose molecules link together. We cannot digest cellulose. And so you may have heard of that you need a certain amount of fiber in your diet. We use fiber to help pass foods along through our digestive system. And so it's important to eat a good amount of fiber. It says some microbes use enzymes to digest cellulose. So microbes, these are little tiny small bacteria. Cows, let's think about cows for a second. Cows, what do they eat all day, every day?
Grass.
Grass, okay, and they're not able to digest this grass. Okay? And that's a problem. So here's what they do. They send their grass into one stomach where it is introduced to a bunch of microbes, a bunch of little tiny bacteria. These bacteria are able to digest cellulose. So these bacteria start breaking down the grass into these polysaccharides called cellulose, get broken down into glucose. The cow, you've ever seen a cow? They'll go lay down sometimes in the afternoon, and they keep burping and then they're chewing. They're burping and they're chewing. Guess what? They are pulling back all of that grass out of one stomach. They chew it and they send it to another stomach after the microbes have helped to break it down and digest it. Okay? And so we're the same way. If we're not able to break down cellulose when we eat it, if you've ever eaten things like broccoli or whatever, that will pass through as fiber. And you may even, this is gross, but you may even get gas if you eat a bunch of fiber type foods because the microbes will break it down, and in the process of doing that, they're going to release some gases. Okay? It says many herbivores, from cows to termites, have symbiotic relationships with these microbes. So cows are very thankful for these bacteria that help to break down grass for them. All right, number 4. This is our 4th big polysaccharide. That CH makes a K sound. So it's called chitin. Chitin. And this is a big structural polysaccharide that's found in the exoskeleton of arthropods. Also found in ants or crickets or grasshoppers, that hard, crunchy exoskeleton. Arthropods, spiders will have that hard, crunchy skeleton. And then crabs, shrimp, crawfish, and that hard, crunchy shell is made from chitin. It says chitin provides structural support for the cell walls of many fungi. So it's also found in the cell wall of fungi like mushrooms. Okay? And it is made from sugars, the big giant sugar. You've ever been on a picnic? You had set your food out. You may go do something for a minute and you come back. What are you going to see everywhere? Ants. Okay, they're going to come after your sweet, sugary foods because they are going to need glucose to help build their chitin. Okay, so they're going to come after your supplies. Where are we? Y'all give me just a second. Let me see how far I want to go. I think we'll I usually do this before I start to remember where I'm at and what I want to do. What I do. Tertiary. We'll go ahead. We'll cover lipids today. We'll have a shorter day on Thursday. How about that? Okay. So lipids. Let's start with lipids. If I can find it. Where were we? There we go. Okay. So we're going to go ahead and cover lipids. This will be section 5.3. And so we will cover proteins and nucleic acids when we come back on Thursday. Okay? So, back out again. Okay, so we're leaving carbohydrates. We're done with carbohydrates. That's our first big macromolecule. That was the sugars. So this is number 2. This is our second big macromolecule, and we're going to call this lipids. Remember I said it's a little bit different. So it's not technically a true polymer, but it's still made up of a repeating unit that we're going to talk about. Okay? Okay, so it says the unifying feature of lipids is that they mix poorly, if at all, with water. And so they are not going to like water. We're going to say they are hydrophobic. We're going to call them the fats. We're also going to use lipids for building phospholipids, which are going to be found in our cell membranes. And we're going to need a certain amount of good fats to build steroids. Two important steroids we're going to talk about are estrogen and testosterone. Are steroid hormones that give us female and male characteristics. Okay, make sure if you didn't write that down when I said it, they are hydrophobic. They mix poorly, if at all, with water. They're going to run away from water. We know that if we were to take some type of oil and water, mix them together, we would come back and there would be a separation between the two. Okay, oil is less dense, so it would be floating on the top. Okay, so we're going to see that there's good fats and bad fats. They're going to be made, all fats, they're constructed of two types of smaller molecules. The first portion is a glycerol, and the second portion is called a fatty acid. And in our example over here, we're going to talk about the fat molecule that we call a triglyceride. Tri from math means what? Three. So let's look at this one down here in the bottom. This gray portion of the molecule right here in our picture, that would be the glycerol. So it's going to be made of a glycerol. There's the first half. And then we've got 1, 2, 3, these yellow portions. Y'all see these carbons that are linked forming these tails? We call those tails a hydrocarbon. It's nothing but a bunch of carbons linked together with some hydrogen sticking off of it. We talked about hydrocarbons a little bit in chapter 4 when we were talking about carbon and it being able to form these long chains. There's lots of energy in these hydrocarbon tails. So these tails, this would be the glycerol portion right here. And then we've got these 1, 2, 3 fatty acid tails. Okay? And so that's what our fat molecules look like if we were to diagram them out and show their molecular structure. It says glycerol is a 3-carbon alcohol with a hydroxyl group attached, and then the fatty acid just has that carboxyl group attached to a long carbon skeleton. And that long carbon skeleton is going to have lots of energy inside if we start breaking it down. And so that's why we say fats are good for long-term, write this down, long-term energy storage. And we know that animals like a polar bear or a seal or sea lion that's going to have to survive a harsh winter, we know they're going to eat lots of fatty foods. They're going to even be wasteful at some point whenever they're getting fish and stuff. They're going to eat certain portions of the fish because they know it contains lots of fat, so they can store these fats up and get really, really big. Sometimes animals like bears will do this before they go into hibernation. Okay, so long-term energy storage comes from our fats. We ever seen these shows like Alone or where they go out in the wilderness and they try to survive? And they'll start out and then you'll see a picture of them like 30 or 40 days later. Do they still have fat on their body? No, they'll be kind of like a bag of bones. Okay, so their fat reserves get used up very, very quickly. Okay? It says fats separate from water because water molecules hydrogen bond to each other and exclude the fats. Okay, remember we talked about water bonding with water cohesion. Doesn't allow the fats to bond. In a fat, 3 fatty acids are joined to a glycerol. We named that just a minute ago on that last slide. We said it's called a triglyceride. The fatty acids in the fat can be all the same, or there can be different types of tails. Okay, that's not real important, not anything you need to know for a test. Fatty acids vary in length or the number of carbons and in the number and location of double bonds. And so this is where it's going to matter. We've got 2 types, titled this, 2 types of fats. 2 types of fats. Number 1, these are going to be the saturated fatty acids, saturated fats. They are going to have no double bonds. That's important. Write it down. No double bonds. I'm going to come back and show you in a picture in just a minute what I mean. Because they have no double bonds, they are going to be solid at room temp. So solid at room temperature. Give yourself an example. Here's an example of a saturated fat, and that would be butter. Yummy butter. Well, you like butter, right? You ever go to O'Charlie's and get those rolls, and they bring out that little tub of butter? You got a whole basket of rolls, but you want to put that whole little tub on like one roll. And you're like, can I get some more butter, please? All right, there's a problem with this because these fats that are solid at room temp are not healthy for us, okay? Because they're tails. Look at these tails. We're going to look at this molecule right here. Do you see how these fatty acid tails, we got 1, 2, 3 chains of these carbons, and there's no double bonds. And so their tails are perfectly straight. And what happens is, if you've got multiple fats with all straight tails, they can stack perfectly on top of each other. And that allows these types of fats to clog your arteries. So if they stick to the side of your blood vessel and another one comes along and it sticks to it, their tails perfectly match up and they will clog your arteries. This causes heart disease, can cause a clogged artery, and it can kill you, can cause you to have a heart attack. Okay? So saturated fats are not good for us. Our second one is an unsaturated fat. Go back to saturated real quick. We're going to add one more thing. Saturated fats, put this in there. They're solid at room temp, and they come from animal fats. Animal fats. We said butter was an example. We know that we get that from cows, right? Have y'all ever cooked or seen somebody cooking bacon in a pan? And you come back, you set your pan off to the side so your grease can cool down. You come back maybe in an hour or so. What do you notice about the grease in the pan? It turns white, it gets solid. Okay, that's animal fats. They're saturated, they're solid at room temp. You ever seen my mama used to do this when I was a little girl. She had this container, it was like a little canister, used to sit on her stove. And so anytime she cooked something like bacon, she would pour that bacon grease, leftover bacon grease, in that little canister. And then later on, if she was cooking something else, she may take a spoon of that bacon grease and put it over in the green beans, if she's cooking green beans. It makes the green beans taste great, right? What do we normally see in green beans when we're eating them? Bacon, okay? And they taste great, but what are we doing? We're adding saturated, unhealthy fats to a vegetable that was going to be healthy for us. So we have to be careful with unsaturated fats. Okay, too many of them are not a good thing. Here's what we do need. Let's pick back up where we left off here. Unsaturated fats, they are going to have one or more double bonds in their tails. Okay, so one or more double bonds. Come over here and let's look at our picture of this molecule of an unsaturated fat. We have our glycerol portion. My screen is acting crazy here. There's the glycerol portion, and we have 1, 2, 3 fatty acid tails. But if we look at this third tail, everybody see the third tail? Right here is what we call a double bond. And when there's double bonds between those carbons and those hydrocarbon tails, it causes the tails to get kinks in them. And they're not able to stack up on top of each other. And because of their molecular structure, unsaturated fatty acids or fatty unsaturated fats will be liquid at room temp. I'll write this down. They're liquid at room temp. They are healthier for you, and they are going to come from vegetable fats. Those come from vegetable fats. Okay? And so give yourself an example. In our picture right here, they have olive oil. And you've probably heard or seen on TV when they're cooking with the extra virgin olive oil instead of butter. They're trying to be a little more health conscious as far as using vegetable fats instead of the animal fats. So, next time you go to bake a cake, butter's going to make it taste great, but what's a healthier version?
Oil.
Now, we could use olive oil, or we could use that liquid vegetable oil would be better. So anything that is liquid at room temp would be better than butter. Okay?
Miss Lala.
Uh-huh.
So a lot of the time I'll see things on the back of like food labels and it says polyunsaturated fatty acid. Like what is polyunsaturated fat?
That's the same thing. They're just larger saturated tails. And we used to have something you might have seen, hydrogenated foods. We don't do that anymore, but we used to take unsaturated fats and hydrogenate them. We would add extra hydrogens to them, and it would make them become saturated. And so we realized that all of these saturated and polysaturated or polyunsaturated fats, the saturated ones are not good for us. Okay, so the back of the food label, you can read, if you go get a bag of chips, it'll tell you how many saturated or how many unsaturated, but when we add poly to it, that just means many. So many saturated fats or unsaturated fats. It says fats made from saturated fatty acids are called saturated fats. They're solid. Animal fats are going to be the saturated. Remember, plants. Fish is another place where you can get the unsaturated fats. If you've ever heard of the omega-3 fatty acids, those are good for you also. You get that from eating fish. You can actually go to the vitamin store or Walmart and buy a little pill, fish oil pill, and take that. Those will help reduce heart disease. This hydrogenation I talked about, that's where we convert unsaturated to saturated. They got rid of that years ago. It says these trans fats may contribute more than saturated fats to cardiovascular disease. So it's kind of outlawed. We don't hydrogenate oils anymore. We said that the major function of fat was for long-term energy storage. Humans and other mammals will store it up in what we call adipose cells. So adipose cells are fat cells. Now, we also use adipose tissue to cushion vital organs and insulate the body. So a certain amount of adipose cells we need. Each one of our organs is kind of in this little individual sac and kind of holds those organs in place. So we do need to eat a certain amount of good fats. And remember, we also said that fats are going to be used for building your phospholipids, which are going to be found in every single cell membrane of your body, okay, every single cell. And for steroids. So phospholipids, here's another reason. Get this, what is a phospholipid? You're going to have two fatty acid tails. That's going to make the tail portion. And we're going to have a phosphate group. We're going to connect those to a glycerol, just like we did in the triglyceride a while ago. But this time we're going to build what we call a phospholipid. And so let's look at the molecule. Here's the phosphate group right here. Here's the glycerol. And here's 1, 2 fatty acid tails. And this is what your phospholipids are going to look like. They're going to form your cell membranes. And then we're going to put them together in what we call a phospholipid bilayer. Bi means 2. We're going to have 2 layers of these phospholipids. And so down here in this picture, we've got the heads with the tails pointing inward. Okay? Kind of forming a little layer. And then on the other side, we've got the heads with the tails pointing inwards also. And so we're going to have this double layer of these phospholipids that are connected to each other that's going to form what we call our phospholipid bilayer. It's going to protect our cells. And we're going to have a chapter where we come back and talk about your cell membranes and how these phospholipids work to allow certain molecules in and then prevent certain molecules from entering into the cell. Okay? The heads up here, these hydrophilic heads, they're going to be positive. These tails are going to be negative, and they're going to be hydrophobic. And so they are not going to want to bond with water. And so water will not be able to stay on this inner portion of your cell membranes. It's going to have to kind of pass on through. And so phospholipids are found in every single cell in your body. You do need a certain amount of good fats. Let everybody catch up real quick. All right, we good? It says when phospholipids are added to water, they self-assemble into double layer sheets called bilayers. At the surface of the cell, the phospholipids are arranged in a bilayer with the hydrophobic tails pointing toward the interior. So this would be the inside. This says extracellular, so this would be pointing out of the cell. This is intracellular, so this would be the inside of the cell. So if we had a whole entire cell, we would have this double layer of these phospholipids all the way around, okay? It forms the boundary between the cell and its environment. Another reason we need fat, so this is this under lipids, steroids. So far we've talked about phospholipids and now steroids. So steroids, they are lipids that are characterized by a carbon skeleton consisting of four fused rings. And so down here, there's 1, 2, 3, 4 fused rings in a lipid molecule. One example of a steroid that we use for many purposes is cholesterol. So you can put this as an example of a steroid is cholesterol. This is a component that we add to animal cell membranes. So if helps to keep our cells fluid. We're able to move around, right? So if you grab your skin, it's got movement to it. We're not locked in place like a plant. And so in the cell membrane of every single cell that we have cholesterol kind of embedded in the membrane to give it some fluidity. They're also a precursor, which means this molecule is a base unit that is used to build some other steroids called estrogen and testosterone. Okay? It says a high level of cholesterol in the blood may contribute to cardiovascular disease. So if you've ever heard of somebody say they have high cholesterol, that means they have a high amount of fats flowing in their blood. Not a good thing. And we get that from eating too many animal fats. So, too much red meat would be a bad thing. Too much eggs. We can actually eat too many eggs and get a lot of cholesterol.
High cholesterol is genetic. How is it genetic?
It's it's not technically just genetic. I mean, it can be kind of genetic, and that would be related back to how you process or synthesize the cholesterol, maybe based off of your metabolism and how you break it down, I guess, would make some of it genetic. But it wouldn't necessarily be just genetic, but it could be genetic. And then some of it is like based off of how you live, and like if you live in a house where people are always frying foods and eating a lot of unhealthy type foods, then as a kid you're going to grow up in that environment. You may even do the same thing, so that could also tie it to, you know, a particular family. But I will look that up and see. Good, that's a good point. Okay. All right. So we wrote down estrogen and testosterone, right? I cleared it off, but this portion right here of a cholesterol, that's like the base unit. And if we were to pull up a picture, I don't have one on here, but if we pull up a picture of estrogen and a picture of testosterone, those same 4 rings are used as the base unit to build estrogen and testosterone. And so those are steroid hormones that are made from your fats that you eat, okay? We're gonna pick up with proteins. Y'all write this down where y'all can remind me on Thursday. We'll pick up with proteins. Proteins will be our third macromolecule that we're gonna talk about when we come on Thursday, okay? There are a couple of extra little videos that are in your modules in Canvas that you may want to kind of watch on your own. We're not really going to have time to watch those in class, but there's a couple of extra little videos in there. Okay? All right. Does anybody have any questions before we go?
But will you teach biology 2 next semester?
Do I teach biology too? Is that what you said?
Yeah.
Unfortunately not. That's a question I get asked every semester, but unfortunately I do not. Here at Poplarville, Miss Lee teaches biology 2, and I think Miss Warden teaches biology 2 at the Forest County campus. But I don't teach biology 2. Sorry.
Bye, we love you.
What did he say?
Said why? We love you.
Oh, okay. Well, that's a good thing. I don't always get told that everybody loves me, but I try to be a good teacher. So if y'all do have questions about something or need something, just let me know, okay? All right, everybody's still reading your book, right?
Yes, ma'am.
Okay. In the next couple of weeks, we may start talking about some of the first couple of chapters. So you're not going to know what we're talking about if you're not kind of starting in your book, at least, okay? All right, I will see y'all Thursday.
All right, have a good day.
Bye, y'all too.
Miss Lawler, I do have a quick question.
Okay?
I submitted my my topic a few weeks ago.
Okay.
A couple more options.
Let me
Look at it and let me know.
Yeah, let me see if I wrote it down. I may have written it down and just forgot to email you back. Let's see. I think that's in my other folder, Kristen. I will check I'll go back and, did you submit it under the comments of that section?
Yes, ma'am.
Okay, I'll go back and check it and I'll email you back and let you know, okay?
Okay, thank you.
All right, y'all good? All right, I'll see you
Yes,
on
ma'am.
Thursday.
See you Thursday.
See you then.